I'm Christopher Winrow, Vice President of Translational Medicine and Development Program Lead at Cyclerion Therapeutics. Today, we're pleased to share positive top-line clinical data from our Phase 2A clinical study of CY6463 for the potential treatment of mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episodes, also known as MELAS. At Cyclerion, we're on a mission to develop treatments that restore cognitive function. Our approach centers on the nitric oxide, soluble guanylate cyclase, cyclic GMP pathway, which plays a central role in neurodegenerative and neuropsychiatric diseases and has been linked to mitochondrial biogenesis and function. Our lead program, CY6463, is a positive allosteric modulator of soluble guanylate cyclase, or SGC, which amplifies endogenous nitric oxide signaling. It is the first stimulator of SGC capable of crossing the blood-brain barrier and modulating this pathway in the central nervous system. The goal of the study we're here to discuss today was to evaluate the safety, tolerability, and multidimensional pharmacology of CY6463 in patients with MELAS, a debilitating mitochondrial disease that severely impacts multiple organs, including the central nervous system. MELAS significantly impacts patients' life expectancy, quality of life, and the ability to live independently. And while it's one of the most common and severe mitochondrial diseases affecting up to 20,000 patients in the United States, there are no currently approved therapies. Dr. Chad Glasser, Director of Clinical Research, will walk through the study design and top-line data, which demonstrate that treatment with CY6463 was well tolerated and had an impact on important aspects of MELAS disease following 29 days of treatment. We believe these data support further development of CY6463 and suggest that it has the potential to be an important treatment option for patients. And now I'll turn things over to Chad.
Thank you, Chris. This trial was an open-label, single-arm study of our oral once-daily SGC stimulator, CY6463, in adults aged 18 or older with CNS symptoms consistent with MELAS. The primary objective of the study was to assess the safety and tolerability of a 15 milligram once-daily oral dose of CY6463 over the course of 29 days. The study also provided us an opportunity to explore the pharmacokinetic profile of CY6463 and its effect on a range of disease-relevant pharmacodynamic endpoints, with the goal of identifying biomarkers that we can carry forward into additional studies in MELAS and potentially other mitochondrial diseases with CNS symptoms. The pharmacodynamic endpoints included biomarkers of mitochondrial function, including lactate and GDF15, a broad range of inflammatory biomarkers, cerebral blood flow using arterial spin labeling, and finally, functional connectivity using fMRI. The study enrolled a total of 8 participants representing various levels of disease severity, with 6 of the 8, or 75%, receiving concurrent nitric oxide precursors, arginine or citrulline, which are often considered standard of care for many of these patients. Data from the study confirmed CY6463's strong safety profile. It was well tolerated. There were no reports of serious adverse events or treatment discontinuations due to adverse events. The pharmacokinetic profile and concentrations of CY6463 in the cerebral spinal fluid and in the plasma were consistent with what we've observed in phase 1 studies in healthy participants. Additionally, we observed consistent trends towards improvement across multiple disease-relevant biomarkers. We observed improvements in biomarkers of mitochondrial function that we know are impacted in MELAS, including lactate and GDF15. The changes in these biomarkers also tended to correlate with one another, meaning that they changed similarly within a patient. These reductions also correlated with plasma concentrations of CY6463, meaning that the higher the exposure at the end of treatment, the more pronounced the reduction. These correlations increase our confidence that these are drug-related effects. Improvements were observed across a broad panel of inflammatory biomarkers evaluated in our study. Similar to the biomarkers of mitochondrial function, changes in many of these inflammatory biomarkers also correlated with plasma concentrations of CY6463 at the end of treatment. Increases in cerebral blood flow were also observed across all the regions that we analyzed, and these improvements even correlated with clinical improvements as assessed by the Patient Global Impression of Change scale. Consistent with improvements in blood flow, we also observed increases in functional connectivity between neural networks associated with executive function and sensory motor processing via resting state fMRI, as well as increased voxel activation within the occipital region following presentation of a visual stimulus compared to baseline. I think what excites us most about these results is the fact that we were able to observe consistent effects across multiple objective biomarkers and across multiple disease domains at such a small and heterogeneous sample. These data also confirm the pharmacology that we've established preclinically, which increases our confidence in the treatment effect and will inform the strategy of our next phase study. We'd of course like to thank the patients, the families, clinical staff, and caregivers that have contributed to this important work, and also the study team at Cycleria.
Thanks for sharing the details of the study, Chad. As you can imagine, we're very pleased with the clinical data generated from the study, including the safety and tolerability of CY6463, in addition to the positive impacts observed across multiple objective biomarkers. We believe this data support the further development of CY6463 in MELAS. These findings are exciting, and as we think about the potential of the mechanism in mitochondrial disease, we also think more broadly about how the effects of mitochondria are likely to underlie dozens of other CNS diseases, including schizophrenia and Alzheimer's disease, which are also associated with mitochondrial dysfunction. To view the data presentation, please visit the Publications and Presentations page on www.cyclerion.com.